Welcome back from the break to Session 2, Oral PPK for Evaluating the Impact of Food on BE. This session has 3 speakers. The first speaker, Dr. Abdullah Shoyaib, is RS Fellow serving in the Division of Quantitative Methods and Modeling, Office of Research and Standards, OGD. Currently, Dr. Shoyaib is working on quantitative methods development, such as PBPK and population-based pharmacokinetic model for assessing the impact of food and excipients on bioavailability and bioequivalence of oral dosage forms. The second speaker, Dr. Arian Emami Riedmaier, is a senior principal scientist and PBPK lead at Bristol Myers Squibb. Dr. Riedmaier is a subject matter expert in translational modeling and mechanistic ADME and has chaired an IQ consortium working group focusing on assessing the predictability of PBPK models for understanding food effect. The third speaker, Anita Kumar, MPharm MS Science, is a vice president in R&D at Amneal, holding a broad spectrum of scientific and management experience from industry. Dr. Kumar oversees a comprehensive product development pipeline. Following the 3 presentations, there will be a panel discussion. The moderators are Dr. Partha and Neil Parrot. Dr. Roy is the director of the Office of Bioequivalence, OGD. Dr. Roy is a recognized senior clinical regulatory strategist and proven business leader with 21 years of drug development experience in both US FDA and industry involving new drugs, novel biologics, generics, and biosimilars. Neil Parrot works in the Translational Modeling and Simulation Group, which is part of the Pharmaceutical Sciences Department of Roche Pharmaceutical Research and Early Development in Basel, Switzerland, and specializes in PBPK modeling and its application to Roche projects from early discovery to the market. Let's listen to the presentations. Thank you.
Good afternoon, everyone. My name is Abdullah Shoyaib, and in this part of today's workshop, I will be discussing the development and validation of physiological-based pharmacokinetic model, also known as PBPK model, for predicting food impact on bioequivalence assessment. This is my disclaimer. To begin with, I would like to give you an overview of PBPK model development and its implications. For PBPK model development, our whole body is divided into different tissue compartments, as you can see in this figure. Movement of the administered drug through these compartments is quantitatively modeled using a series of differential equations, and this mathematical model is parameterized using true physiological variables such as tissue, volume, and blood flow. PBPK model mechanistically combines both drug properties and mechanism of drug absorption, distribution, metabolism, as well as elimination, while predicting the plasma profile of the administered drug in a particular site of interest, such as plasma profile. Later, this predicted plasma profile is compared with the clinically observed plasma profile, and if necessary, appropriate parameter optimization is conducted to get the best fitted model. Finally, validation of the best fitted model is conducted using independent PK data, which were not used for model development and optimization purpose. Due to the inherited mechanistic prediction capability of PBPK model, we have observed an exponential growth in application of PBPK model for studying different clinical situations, such as drug-drug interaction study, optimization of dose for pediatric population, as well as renally or hepatically impaired population. Additionally, PBPK model has also been used for setting clinically relevant specification as well as for assessing food impact and bioavailable potential. These mechanistic PBPK models can be used to model the mechanism of drug absorption for different routes of drug administration, such as oral, dermal, pulmonary, or ocular route of drug administration. However, among the marketed drug products, about 62% are intended for oral administration. Noticeably, around 40% of these orally administered drugs show significant food impact, meaning taking the drug with food impacts the pharmacokinetics of the orally administered drug. Recently, Pvk model has been used to predict the food impact and to conduct the bioequivalency study using virtual population, which is also known as virtual bioequivalency study, abbreviated as VBE. Considering the mechanistic potential of PBPK model and their implications, the aim of this study was to develop a PBPK model for Acyclovir immediate release tablet to assess the impact of food on bioequivalency assessment using virtual population. To talk about Acyclovir, it is an antiviral drug which is effective against herpes simplex virus. This drug is slightly soluble in water and is available in the market as 200, 400, and 800 milligram doses of IR tablets. It has an absolute bioavailability of 10 to 30%, which is due to the low intestinal permeability of this drug. Here, I'll briefly point out the model development steps for our Acyclovir PBPK model. We started with the data collection related to the Acyclovir physicochemical properties and pharmacokinetic studies. To begin the model development process, first we developed Acyclovir PBPK model for IV dose, followed by validation of IPPBK model to get the disposition parameters. Next, we use these disposition parameters to develop oral PBPK model of Acyclovir under fasting conditions, followed by oral PBPK model validation under fasting conditions. Then we developed the Acyclovir oral PBPK model under fed condition and validated the model with the observed PK data under fed condition. Finally, a virtual bioequivalency study was conducted using virtual population under both fasting and fed conditions. To develop the Acyclovir PBPK model for IV administration, we have used Acyclovir physicochemical properties collected from literature and predicted the plasma profile of Acyclovir after IV infusion of 2.5 milligram per kg of Acyclovir. This slide shows the PBPK model predicted plasma profile and observed plasma profile for Acyclovir 2.5 milligram per kg IV infusion along with the prediction error. Here, the blue solid curved line represents the PBPK model predicted plasma profile, and the blue small box represents the observed PK data along with the standard bar. And the table on the right side represents the prediction error. Next, we also predicted the plasma profile for 5 milligram per kg Acyclovir IV infusion without changing any model parameters except Acyclovir dose for validation purpose. As you can see, the IV PBPK model predicted the plasma profile of Acyclovir 5 milligram per kg dose well with a prediction error less than 20%. From this IV PBPK model, we get the disposition parameters of Acyclovir such as volume of distribution and clearance. In order to develop Acyclovir PBPK model for oral IR tablet or immediate release tablet, disposition parameters from IV PBPK model was used along with the addition of intestinal permeability parameters. This is how our oral PBPK model predicted plasma profile for 200 milligram Acyclovir immediate release tablet looks like. Comparison of the predicted model profile with the observed PK data of 200 milligram Acyclovir IR tablet shows a good model prediction capacity of our oral PBPK model with a prediction error less than 20%. Later, we increased the dose from 200 milligram to 400 milligram immediate release tablet and compared the predicted plasma profile with the observed pharmacokinetic data for 400 milligram IR tablet from a separate study. As you can see, for 400 milligram dose also, our PBPK model predicted the plasma profile well. Finally, we predicted the plasma profile of Acyclovir 800 milligram IR tablet and compared that with the observed pharmacokinetic profile of 800 milligram immediate release tablet. This ensured the robustness of our developed oral PBPK model. For PBPK model validation purpose, we compared the model predicted plasma profile for 400 milligram IR tablet with an independent PK study, which was not used during the model development process. As you can see that the model was able to predict the observed data well. Similarly, we have also validated our developed PBPK model for 800 milligram IR tablet using internal ANDA data, which are not shown in this presentation. Additionally, we have also conducted population simulation for 400 milligram Acyclovir immediate release tablet. Here in the population simulation, the black solid line represents the mean plasma profile of the simulated population, whereas the green solid area represents the 90% confidence interval of the simulated population. The yellow shaded region with the dots represents the 90% probability of the predicted plasma profile for the simulated virtual population. Similarly, these are the population simulations for 200 and 400 immediate release tablets under fasting condition. And this table summarizes the prediction performance of the Acyclovir oral PBPK model for 200, 400, and 800 milligram IR tablet under fasting condition, along with the prediction error. Our next task was to develop the Acyclovir oral PBPK model for fed condition. As we all may know, that food intake has substantial impact on physiology and local environment of gastrointestinal tract. For example, food intake increases the liver and portal blood circulation. In stomach, food increases the pH and the volume of the stomach, but delays the emptying of the gastric stomach. Food intake also impacts the bile salt release from gallbladder. Additionally, food intake can also impact the local condition of small intestine by increasing the bile salt concentration, motility, and viscosity of small intestine. In colon, food intake can increase the buffering capacity and osmolarity. Hence, for developing oral PBPK model for fed condition, these physiological changes need to be accounted as they may impact the dissolution, precipitation, as well as absorption of orally administered drugs. Here, from the structure of Acyclovir, we can see that Acyclovir is an ampholytic drug which has an ionizable basic and an ionizable acidic functional group. Thus, Acyclovir can be ionized at different pH, which can ultimately impact the solubility of Acyclovir, as you can see in this figure. Similarly, food intake may affect the solubility and precipitation of Acyclovir due to the change in pH in stomach and small intestine, which can further be impacted by the change of bile salt concentration in small intestine due to food intake. Collectively, these changes can impact the pharmacokinetic of Acyclovir after oral administration. Under fasting condition, this was the PBPK model predicted plasma profile of Acyclovir 800 mg IR tablet, as you have seen before. Now, when we change the physiology of our PBPK model from fasting to fed condition, this is how the PBPK model predicted plasma profile looks like under fed condition for 800 mg IR tablet of Acyclovir. As you can see from the graph that there is a change in Acyclovir PBPK model predicted Cmax, Tmax, and AUC under fed condition compared to the fasting condition. Comparison of this predicted plasma profile with the observed PK data under fed condition shows that the model is capable of predicting the impact of food on pharmacokinetic of Acyclovir with a prediction error less than 20%. These two graphs shows the population simulation of Acyclovir 800 mg IR tablet under fasting and fed condition. From all this PBPK model predicted plasma profile under both fasting and fed condition, we have seen that our model was able to predict the plasma profile well under fasting and fed condition, and hence this model can be used to conduct virtual bioequivalency study under both fasting and fed conditions. Thus, our final task was to conduct virtual bioequivalency study of Acyclovir 800 mg IR tablet under fasting and fed condition. For this purpose, we have used in vitro dissolution data for both reference and test products from ANDA data as our PBPK model input. This slide shows the bioequivalency result under fasting condition. As you can see that the virtual population simulation shows bioequivalency between the test and the reference product, which is similar to the ANDA bioequivalency result. Finally, we conducted the virtual bioequivalency study under fed condition, and our simulation results show that the test and the reference tablets are bioequivalent under fed condition as well, which is similar to the ANDA fed bioequivalency results. In summary, Here we have shown the development and the validation of Acyclovir PBPK model for immediate release tablet. The model was validated using PK data from both published literatures as well as approved ANDS data. This validated model was capable of predicting food impact on Acyclovir pharmacokinetics data. Finally, the Acyclovir PBPK model was used to conduct virtual bioequivalency study under both fasting and fed conditions. With that, I would like to thank my mentor, Dr. Fang for her consistent support and valuable guidance. I also appreciate the cooperation of other oral PPK team members, Dr. Yusuf Musa and Dr. Yishan Cheng. Finally, I truly appreciate the valuable suggestions and directions from DQMM Division Director, Dr. Liang Zhao, and Deputy Director, Dr. Lucy Fang for this project. And thank you everyone for listening.
Hello, and thank you for attending my presentation entitled Predicting the Power of Food using PBPK Modeling to Predict Food Effect. I would also like to thank the FDA and the Center for Research on Complex Generics for the invitation to speak at this workshop. Before I start, I'd like to acknowledge that this presentation was developed with the support of the International Consortium for Innovation and Quality in Pharmaceutical Development, or IQ. IQ is a not-for-profit organization of pharmaceutical and biotechnology companies with a mission of advancing science and technology to augment the capability of member companies to develop transformational solutions that benefit patients, regulators, and the broader research and development community. The IQ working group that performed the analysis that I will discuss in this presentation consisted of 21 individuals representing 12 member companies that are shown here. Dr. Neil Parrot and I were the co-chairs for this working group. The work that I will present here has been published in 3 different manuscripts that I'm showing on this slide. If you're interested in digging a little deeper into our analysis and conclusions, I would refer you to these publications. As many of you know, due to changes in GI physiology in the presence of food, absorption of orally administered drugs can be affected when taken with a meal, and food effect and bioavailability studies are usually conducted to support NDAs and label recommendations. In early discovery and development, various tools are used to predict the potential or the risk for food effect. These include studies in preclinical species, in vitro biopharmaceutics models, as well as the use of in silico models such as PBPK. Once in clinical development, clinical food effect studies are conducted and can be then used to verify food effect predictions. The verified food effect predictions can then be used to extrapolate food effect to novel formulations, other doses, as well as special populations. Given the complex nature of food effect, an integrated approach is required. Physiologically based absorption models have emerged as a key platform for the support of food effect predictions. Various publications from industry, including an IQ paper that was published in 2015, have shown high to moderate confidence for predicting food effect using PBPK for compounds where intestinal transporters don't play a key role. However, a retrospective analysis that was published in 2018 concluded that we're not there yet for several compound classes and that more in-depth analysis is lacking. In the meantime, while BCS classification could serve as a generalization of drug property, appropriately verified physiologically relevant models can provide a more powerful assessment of drug property in combination with pharmacokinetic and physiological considerations. At the start of this initiative, there were no publications available that were able to assess the ability of PBPK models to predict absorption and food effect using a consistent and prospective approach. We formed a cross-functional team of formulation scientists and modelers from 12 pharmaceutical companies and worked to establish a consistent workflow for modeling with standardized input data, agreed-upon principles, decision trees, and data generation methodology. We also ensured that there was appropriate verification of models prior to a food effect prediction and/or recommendation. Our vision was to have a well-conducted and published verification study of food effect prediction using PBPK, which would aid to understand modeling applications and highlight cases where we would have high and moderate confidence versus low confidence in predicting food effect. Ultimately, we wanted to provide an aligned industry perspective on cases where modeling may be used in lieu of clinical studies. For this modeling exercise, we used SimSip version 17.1 and GastroPlus version 9.5. All system parameters were left as default and literature values were used for all PhysChem and blood binding parameters. The absorption models were developed mechanistically based on an aligned workflow and decision tree, where all in vitro input data were generated within the working group unless there was literature data available generated using the working group aligned methodology. The distribution and elimination was informed using IVPK data in order to remove any model uncertainty around drug disposition and isolate the verification step absorption component of the model. It's important to note that this is not a recommendation that we're making in this exercise. We just use this approach to reduce uncertainty and focus on the absorption parameters given our resources and timelines for this work. Clinical studies were also clearly defined as training versus verification and test set to avoid any bias. We also excluded compounds that were expected to have high hepatic extraction and compounds where absorption is expected to be limited by active intestinal transport as well as prodrugs. This figure shows a distribution of the compounds that we analyzed. We started off with a list of 200 compounds that we then narrowed down to 30 compounds after applying all of our exclusion criteria. The selected compounds for the final data set covered a range of solubility, permeability, molecular weight, and lipophilicity properties, as you can see in this figure. As I mentioned earlier, all modelers followed a decision tree for building and verifying the models in this exercise. This decision tree is shown on this slide and is described in more detail in our umbrella publication. The 30 compounds that were tested covered a range of BCS classifications and food effect types, and they consisted of 13 compounds with positive food effect, 8 compounds with negative food effect, and 9 compounds with no food effect. The model performance and confidence was evaluated in the context of the stage of drug development. So for example, if a model was built purely bottom-up, it would be more relevant to the discovery stage of drug development, whereas if it was built in a middle-out fashion, then it would be relevant to discovery and the development stage. And the direction and magnitude of food effect was evaluated using a purely bottom-up versus middle-out approach. To provide an overview of the predicted food effect, the rate of correctly identifying the risk or the potential for food effect was very high. We were able to only find 2 examples of false negatives. Similarly, the direction of food effect was accurately predicted for approximately 90% of the compounds without the need for optimization with clinical data, and the magnitude of food effect was predicted with high or moderate confidence for 80% of the compounds. We found that while assigning confidence based on BCS classification might be an oversimplification, when combined with the driving mechanism of food effect, it could provide a novel perspective on our prediction confidence. For example, we found high confidence in PBPK food effect models for all BCS 1 and 3 compounds where a significant contribution of transporter-mediated food effect could be ruled out, as well as for a subset of BCS 2 and 4 compounds where the driving mechanism of food effect could be attributed to changes in solubility in the fed state related to changes in GI physiology. Where the mechanism of food effect is well understood, but the in vitro to in vivo correlation is weak, for example, for compounds that undergo significant precipitation, a middle-out approach could be utilized with higher confidence using a clinical anchor study. Looking at the impact and magnitude of food effect in relation to Cmax and AUC, as well as the profile overlay in general, we were able to predict 15 compounds with high confidence, which was defined as accurately capturing the fasted and fed PK parameters and profile within a 2-fold range of the observed, as well as having a good overlay of the PK profile. Or if the model had been optimized as defined in the decision tree, we were looking to capture the fasted and fed PK parameter and profile within a more conservative range of 0.8 to 1.25. For 8 of the compounds, we had moderate confidence, which meant that we captured the fasted and fed PK parameters and profile following optimization using fasted data within a 2-fold range of observed PK parameters. And for 7 of the compounds, we had low confidence, which meant that we failed to capture the fasted and or fed PK parameters and profile even after optimization as described in the decision tree. It's important to note in these cases that while modeling the low confidence subset of compounds using a broad predefined decision tree around optimization was not really suitable, when we deviated from the general workflow depending on the mechanism of food effect, it did help in some cases to improve the accuracy of the model. In general, we found that prediction success was correlated to the driving mechanism of food effect. So areas of high to moderate confidence were mainly associated with changes in GI luminal fluid and physiology, and you can see that on the figure on the right. And low confidence cases were commonly associated with complex mechanisms or interplay between complex mechanisms for which we don't have standardized in vitro assays, and the model inputs may not be available to characterize that food effect. Some of the main drivers for low confidence in predictions were found to be the presence of salt forms and their effect on the microenvironment pH, changes in the hydrodynamics or viscosity in the presence of food, and buffer species and how representative they were of in vivo solubility. At the end of our exercise, we defined some areas of improvement and some easy wins to increase our confidence in these models. These suggestions are published in our third manuscript by Wagner et al., which is also cited here. We identified two categories for improvement. The first category involves improvements to in vitro methodologies and mainly includes considering the use of more biopredictive media as PBPK input parameters. For example, the use of bicarbonate buffered media to capture the fed state solubility. The pazopanib case study is a good demonstration of this. The second category involves improvements to the Pvk software, and some easy wins were highlighted to be enabling the use of solubility data from media simulating the fed stomach, enabling the use of full salt solubility profile in the Pvk software to capture the common ion effect, and enabling the ability to capture changes in GI physiology over time. So, for example, allowing the simulation of gastric reacidification. Some improvements that are required but may need more research are more biopredictive tools and correlations to capture precipitation kinetics, improved mechanistic hydrodynamic models that allow users to calculate the luminal drug dissolution, more realistic simulation of gastric residence time of formulation or drug, and a better understanding of the food transporter and food enzyme interactions. And this could involve in vitro tools, in vitro to in vivo correlation, and implementation in the PBPK software. In conclusion, this work explored the predictive ability of PBPK models using de novo mechanistic absorption models for 30 compounds, where an aligned predefined in vitro and modeling methodology was utilized, as well as an aligned decision tree for model design, verification, and optimization. These PBPK models predicted food effect modeling for the majority of the compounds with high or moderate confidence, and a smaller subset of compounds showed low confidence. A correlation was observed between the confidence in the model versus the driving mechanism of food effect. Of course, this study was the first step in understanding the prediction success of food effect and its correlation with the mechanisms driving this effect, and future work should focus on further strengthening the validity of these conclusions by expanding this data set to additional compounds. And we should also explore food type and formulation effect on the prediction success of these models.
This presentation discusses the generic industry perspective on the impact of food and formulation in the context of using PBPK modeling as an alternate pathway to evaluate and determine PE. The disclaimers. Oral PBPK modeling and simulation has been well explored mostly by the specialty pharma space. However, it is now emerging on the generic horizons with many proposed applications. Let us look at some of them. Some of the proposed applications are that they aid in deeper understanding of the drug and reference product. The PBPK modeling will promote alternate BE pathways requiring fewer in vivo studies. It will support biowaivers for lower strengths in case of non-proportional formulations. This pathway can support robust clinically relevant criteria for performance-based tests such as dissolution, and of course, it provides pathway for easier post-approval changes and effective life cycle management of a product. Now, let us look at some of the optimal PBPK modeling workflow and the requirements. Ideally, we are looking for drug candidates with well-characterized GI absorption profiles, BCS class 1, 2, or 3 compounds with linear PK across the dose range, compounds without major transporter influence or saturable metabolism mechanisms, and compounds that are likely to demonstrate biopredictive dissolution profiles. The limitations: we are generally looking for compounds where physicochemical attributes and PK profiles are well understood. Some of the limitations are regarding compounds without published intravenous PK data, compounds where absorption is limited by active transport, such as also prodrugs and compounds that exhibit GI instability, drugs showing high first pass or hepatic metabolism, compounds with significant active metabolites, those demonstrating high in vivo variability, and complex modified release systems with drugs of low solubility. To my right is a proposed workflow that I have pulled from the literature published by one of our generic industry colleagues. This is an example of a mechanistic PBPK approach where the API, physicochemical, and PK parameters are fed into the modeling software. The model is built using the PK data of the reference product in the fasted condition. Next, internal and external validation were carried out using the PK profile, the plasma profiles of the product in the fasted of the reference, as well as for the test in the fasted states. Upon this, the simulation of the plasma concentration and PK parameters in the fed state were evaluated by changing the physiology to mimic the fed state in the modeling software. Finally, a virtual bioequivalence trials were carried out for the test and the reference in the fed state, and as also the evaluation of the prediction of the food effect of each formulation in the fed and the fasted state was evaluated against the observed. The evaluation of the prediction for the bioequivalence in the fed state. We will discuss, this is just an example of what can be carried out, and we will discuss some of the results later on in the presentation. Moving on, let us look at the changes in the fed state physiology with associated effect on the drug. Specifically, the effect of increased gastric pH, gastric emptying time, and motility, increased intestinal fluid volumes and different compositions, and the increased blood flow, which are associated with the postprandial states. The fed state physiology has varied effects on the solubility, dissolution, absorption, and stability of the drug compound based upon the drug class. The major impact of the postprandial state is seen on class 2 drugs, which frequently demonstrate increased bioavailability through enhanced solubility and absorption. Some drugs may be negatively impacted through possibly precipitation kinetics or delayed absorption caused due to the longer gastric emptying time and or site-specific absorption. Of course, the formulation factors that do affect the bioavailability are the API. Some of them are API and certain excipients which are incorporated into formulations such as solubilizers and absorption enhancers, which are used to eliminate or modulate the food effect. I will pause on this slide for a brief minute to just take in all the information. Assessing food effect through 3 different approaches. Food has the strongest impact on BCS class 2 compounds with minimal effects on class 1 and reduced AUC for class 3. The in vitro, in vivo, and in silico approaches have different requirements and yield results, but within the cycle, the chosen approach can be a very effective tool for prediction based on the class of drug being studied. Moving on, there may be no known food effects for a particular drug, and this is mainly seen in class 1 drugs with high bioavailability. The test and reference have similar profiles in biorelevant media, and in vitro method should suffice. For other drugs, you can support BE filing with data through literature and some in vitro as well as some PBPK simulation. For positive food effect, there may be multiple reasons for obtaining a positive food effect, and we have seen some in the previous slide. It is critical to characterize the underlying mechanism causing the positive food effect, and this will help us in selection of the alternate approach to establish BE. Positive food effects arising from increased API solubility and dissolution due to physiological changes have high confidence for in silico prediction for class 2 compounds. Moving on, negative food effects are seen in drugs with pH-dependent solubilities and are sometimes aggravated by micellar entrapment. Also, reduced AUC may be caused by delayed gastric emptying and absorption, and may be due to site-specific absorption of certain compounds. Moving on to potential applications in drug development and regulatory actions, trying to discern between the product types for IR products and modified release products, as the areas of interest for each is different. For IR products pertaining to class 2 and class 3, a combination of in vitro and in silico modeling may be used to evaluate virtual BE for different API forms and critical excipients. In silico approach may also be used for modified release products with absorption dependent PK to evaluate multiple prototypes for virtual BE, or to evaluate complex PK metrics such as partial AUC if the dissolution is biopredictive dissolution with absorption driven PK. Continuing through the potential applications from a regulatory perspective, for the IR products, again, there are three potential areas where it may be used. One is, of course, using the alternate BE pathway for drugs with safety concerns such as oncology drugs, immunosuppressants, and the such like, where we could use, based upon the class, we could use either simple in vitro or a combination of the two for class 2 and 3. Again, there is another possibility where we would use the extension of BE waivers, whereas for class 3, where you would use the in silico BE when excipients differ from the reference in quantity, or F2 does not meet due to very rapidly dissolving test of reference as per the guidance. The last one is, of course, use this as a BE waiver for the fed state, where for the class 2 products, a predictive food effect using in vitro, biorelevant dissolution, and PBPK modeling, as well as for a class 3 if we can use it via in silico to demonstrate fed B state BE. For the modified release products, this approach can be used to support clinically relevant dissolution specifications, where you can be a robust specification that may be outside of the conventional plus minus 10% from the mean using in silico modeling approach and multi-stage dissolution data. To illustrate one of the potential applications, I have pulled a published case study where the food effect was simulated on two different formulations containing API in the crystalline and amorphous forms. The modeler presented the simulated and observed plasma profiles for both the test and the reference product in the fasted and the fed states. The workflow for this experiment was described earlier in the presentation. The model was built based upon the published data of the reference product in the fasted state and followed by internal and external validation using the plasma profiles of the reference and the test product in the fasted state. Upon finalization of the model, the fed state simulation was performed using the fed state physiological parameters. And after this, virtual BE was performed for both the test and the reference product, as well as a prediction for the food effect. Coming to the results of this study, the simulated plasma profiles of the test and the reference product in the fed state are quite similar. In fact, the model predicted that the test was bioequivalent to the reference product in the fed state. Subsequent in vivo trials also confirmed that the virtual bioequivalence model was valid. Coming to the next point, for the food effect, the model was able to predict the food effect for both the test and the reference products quite well. The prediction error for all three PK metrics of both the test and the reference product was up to 10%, thus showing that giving us confidence in the predictions. And therefore, I believe this model was quite successful in its intended experiment. We now come to the last slide, which lists the challenges for quantitative modeling in the current scenario. Despite the progresses that have been made so far in this field, there exists an empirical uncertainty in modeling strategy, modeling input parameters, method of data generation, some subjective optimization processes, and robustness. Is a bottom-up approach better, or when should one use a middle-out approach to evaluate the food effect and try to look at virtual PE? It is a resource-intensive process requiring initial investment from the companies without visibility of concrete returns. Definitely, this process requires timely regulatory participation and agreement for model acceptability and conclusions. One of the big challenges will be the lack of guidelines to establish predictive ability of the models for an intended use. Is it a PE of less than 10 is the guideline, or we can look at a 1.2-fold or a 2-fold prediction error for food effect. Then again, a broad applicability is constrained by drug ADME disposition and drug product design, which means that not all drugs will be suitable for using oral PBPK modeling for alternate BE pathways. Also, there are tremendous challenges in determining biorelevant dissolution profiles and methods for complex systems. And in some cases, in order to truly determine a biorelevant profile, there has to be some in vivo data. So the two things are a little bit counterintuitive in sometimes. And of course, there are challenges to incorporate the inter and intra-subject variabilities to mimic in vivo performance. But last not the least, we do believe this is a good pathway, and there is something that generic industry would explore with sufficient support from the regulations. Thank you very much. I would like to just acknowledge my collaborators and thank you for listening to this presentation and the references. Thank you.
Okay, so I guess we will move to our panel discussion now. I just waiting for a few minutes for all the panelists to join us. I'm also happy to say that Dr. Jasko is going to join us. So I'm just trying to see whether Dr. Jasko can join us. Okay, you all look great. Is Dr. Jasko able to join? I just wanted to kind of make sure. Okay, I mean, I can get started. So first of all, thank you, the speakers, for their excellent presentations, and thanks everybody for joining the session and also this panel. I see Dr. Jasko. Thank you. I think what we'll do is two of the speakers are joining the panel. Abdullah will not be able to, and in his place, Dr. Fang is going to join us. So just wanted to introduce all of the other panelists who are not speakers. Dr. Lucy Fang. Lucy is the Deputy Director for the Division of Quantitative Methods and Modeling in the Office of Research and Standards in OGD. Rebeka Jereb is a scientist. Okay, let me repeat again. Dr. Rebeka Jereb. So congratulations on getting your PhD very recently. Is a scientist in clinical development in Sandoz. Rebeka has expertise in PBPK modeling and IV-IVC and IV-IVR, and has developed various PBPK models for regulatory purposes. Dr. Nilufer Tampal, she's the Acting Associate Director for Scientific Quality in OGD's Office of Bioequivalence. In this role, Dr. Tampal develops strategies and oversees implementation of data quality and the scientific integrity of bioequivalence data submitted in ANDAs. Dr. Fang, you already know her, is a senior pharmacologist and scientific lead at the Division of Quantitative Methods and Modeling in the Office of Research and Standards in OGD. And finally, Dr. Yuching Wang is currently the co-lead of PBPK program at Division of Pharmacometrics, Office of Clinical Pharmacology, and the Office of Translational Sciences in CEDAR. Dr. Young reviews IND and NDA applications and evaluate the application of PBPK tools to support regulatory review and policy development. So welcome all of you to the panel. I think what's going to happen is, the good news is that I think we are running a little bit ahead of time, so we will have a little bit more time, which is great. I'm going to start the questions and then hand over to my co-moderator, Neil. So again, so the first question, so what are the considerations for generating biopredictive dissolution data to be incorporated in the PBPK model and to predict food impact on bioequivalence? I know Kimberly addressed some of the challenges with respect to IR in ER products. So let me see. So Fang, would you take that question?
Yeah, sure. Thank you, Dr. Roy, for this question. Generally, we recommend using dissolution data that can correlate with in vivo release under fed condition and predict well the in vivo PK profiles. We know that there are QC dissolution and biorelevant dissolution method. So the biorelevant dissolution method can be used as a starting point for generating your biopredictive dissolution profiles. In some situations, we also see the applicant use QC method, but if you can demonstrate that your in vitro release can correlate with in vivo release under fed condition, that would also be good. Thank you.
Great. Anita, I know whether you want to expand on that or say anything on that.
Yes, thank you. Thank you, Dr. Fang. Yes, one of the things that definitely as a development during the development of a product, the PK characteristics of the drug as well as the product are very crucial in helping us to develop a biopredictive dissolution. In terms of what is the pH, is the pH independent solubility of the drug, any precipitation of the drug across the pH effects, and what is the drug release mechanism from the product, as well as what could be the possible pH dependent release profile of the drug. And what we always want to try and look at is we try to use a simplistic approach. Would start out based upon, of course, the BCS classification of the drug. We would start out with using a single media or evaluate in multiple medias. Then, of course, we would do a multi-stage dissolution to mimic the GI transit of the product as it moves down the GI tract, as well as, of course, use the different surfactants and bile salts to mimic the fed condition. As one of the presenters did point out, the fed state, mimicking the fed state
in a dissolution medium is still a little bit of challenging because we're not able to totally incorporate all the bicarbonate buffers. But however, I think that's one of the aspects that the industry does work on. And to move the dissolution from a biorelevant into a biopredictive field, definitely we will have to do some simulation and be able to predict to see the predictive systemic exposure is somewhere around plus minus 10%. But those are the few challenges that do exist in getting a biopredictive dissolution media for food effect.
Yeah, because this is an interesting concept because, as regulators, we are always trained to kind of look at the worst case scenario in most of the times. And that's why, you have a risk-based evaluation, in this scenario. So, and so the question is, do you have, the appropriate, the biorelevant or biopredictive dissolution data to inform your model? And extrapolate from there. So I don't know, anybody else want to expand on that or? Yeah, Dr. Tampal?
Yeah, hi. So I was wanting to say, in addition to the biorelevant dissolution medium, right, there are also the mechanistically, when we are using USP paddle 2, can that really reflect the GI, the stress of the GI motility, which may be needed for the disintegration and to develop the disintegration force? Or like if we are thinking about how gastric emptying times can be affecting the dissolution and therefore the bioavailability. So in addition to the medium, the in vitro apparatus that we are using right now, maybe that's the one that we have, but is there any additional research or any additional options that we can be exploring to get a even more biorelevant or biopredictive dissolution data to inform your model then? That was my one comment on this. Thank you.
Thank you. Anybody else? I know, Rebeka, I think you might have, I don't know, did I see you raising your hand?
Yes. I just wanted to comment.
Sure, go ahead.
To be aware of using biorelevant and biopredictive methods, because something can be biorelevant but not biopredictive in view of predicting in vivo differences between test and reference or predicting the food effect. So this can only be tested or seen if we have a biopredictive method, if we have some data, some in vivo data to validate this. So just this short comment.
Right, thank you. Okay, so I will probably go to the next question. So is there a plan to also look into cases where active transport limits absorption? This is a question to Arian.
Thank you for that question. Can you hear me well right now?
Yeah.
Okay. Yeah, I think that's a really good question. It's definitely something that we considered when we were doing our analysis. I think there's several questions that are still outstanding with respect to transporters and food effect. One aspect is just an alignment in terms of the in vitro methodology. Another question is just understanding that in vitro to in vivo correlation and how that in vitro data would ultimately translate to in vivo. And then a third point is just a general lack of clinical examples of this transporter mediated food effect. So for us, the database was still too small to do that kind of analysis at the time that we did the work in this working group. I definitely think we do need more and more examples that can be published for us to actually be able to do this analysis ultimately. So I'm hopeful that in a few years we could get there, but right now I would say that we're not there yet.
Thank you. Neil, I know you are also involved in this one. Is there any comments you may have?
No, from my side, the first comment I'd like to make is, first, thanks for involving me in this very interesting session and these three excellent presentations. And then I'd just like to say, Arian, I think you did a remarkable job to squeeze into 15 minutes such a huge amount of data and results from this IQ study. And I saw that a question came in from the audience actually about one thing that you didn't mention, which was, you know, when we see variability for a drug, we often develop formulations to try to reduce that variability. Did the IQ group look at the effect of formulation on food effect?
Yeah, thanks, Neil, for bringing that up. That's definitely a very important point. It's another point that we discussed at length while doing this work. The work that I presented was really the background work that we wanted to do just to understand where we are in the literature. We have considered looking at formulations, but that is a next step for this working group. So it's something that we didn't systematically look at or evaluate in this in what I presented in this working group right now. However, it's something that we are currently working on as a continuation of the IQ working group. So stay tuned for some of those results very soon, hopefully.
Great, thank you. Okay, so the other question I have, probably the next one, I mean, in case of no IV data is available, I mean, we heard that, you know, there are many cases, and particularly for, you know, a lot of the old, you know, drugs, older drugs maybe, and sometimes the IV data is not available. So can the model still be properly developed and validated and used, for example, you know, for Fed-B prediction? You know, maybe Arian, if you want to take that one, and then maybe go to Rebeka on this one.
Yeah, I think that's again another very good question. It's something that I highlighted that we made sure to exclude cases where we didn't have IV data, but that was really for our own convenience while we were doing this work. In terms of you know, how much confidence we would have if we didn't have IV data, obviously, if we had that data, it would make our lives much easier, and it would improve our confidence. If we don't have that data, in my opinion, we just have to be very cognizant of the in vitro data that we're using and the quality of our in vitro to in vivo extrapolation, and how well we do that and how confident we are. So we just have to have more clinical study, maybe you know, from the oral studies, we just have to have more of those to do the validation and just prove that you know, especially the clearance pathway and the disposition is captured well, so that that's not affecting the absorption aspect of our model. But I do definitely think it's doable. It just requires a larger data set and a stronger in vitro methodology to actually characterize the clearance and disposition pathways.
Great.
Yeah, I had a question actually for both Anita and Abdullah, or for Fang. You both showed in the presentations use of virtual bioequivalence, but I wasn't clear how you predicted the variability in those trials. Wonder you could elucidate a bit on the prediction of the variability in order to simulate those bioequivalence trials. Maybe Fang, would you like to start?
Yeah, thank you, Neil, for the question. Actually, this project was a proof of concept project. We conducted the virtual BE simulations, and when we conducted this, we used the population simulations. So in the population, the variability for the physiological factors are already incorporated in this population. So that's what we use for the variability, covering the variability part. Yeah, thank you.
Thank you. Did you want to say anything, Anita, on this topic?
Yes, so I do believe in this other case study that was demonstrated, the inherent variability that is incorporated from the model, from the software itself, was used for the variability of the populations.
Thank you. Maybe while I'm talking to you, Anita. You mentioned that, you know, there are ideal drugs for this approach, and I was wondering if you could clarify what percentage are considered to be ideal molecules, and is the approach of food effect prediction only viable in the case of these ideal cases, or?
Well, yes, for a percentage wise, I don't think I can tell you exactly the percentage wise, but definitely what I would say for, especially for prediction of food effect, if we use a risk-based approach to choose which molecules or which drugs we would use for that, that would be a class 2 compounds, because I think that we will get the most benefit out of using the PBPK modeling for those kind of compounds. And of course, what we talk about where you have a linear PK relationship, so that we can use it across the whole dose range, whatever the strengths may be. And the limitations, what we also do think is where we have prodrugs. And in case you have some esterases in there, and you want to see the active moiety in the blood, and so example would be something like abiraterone, which is a class 4, where you have the esterase and you have the active in the plasma. In those cases, I think you would have really tough time trying to be able to quantify what you're going to see in the food effect. So from a percentage point, I don't have that. I had no idea, but from a BCS classification, we do know where I think we put our best efforts, we will get good results.
Thank you.
Yeah, just a quick comment and quick. I wanted to make sure Dr. Jasko gets an opportunity to. Sorry, if you have any questions, comments, please let us know. I know you probably have some questions on the chat, but if you have any questions, please chime in. I don't know whether you are in mute. I can see that.
There it goes. It says I'm unmuted. Okay, I have some comments. I'm very impressed by how well the software has worked and the examples provided for assessing food effects. I would never have envisioned that some of this would work so well. On the other hand, it was not always clear what mechanisms were being invoked as the primary ones for determining whether there was an increase or decrease in the food effect. I think it was clear with the first presentation that with Acyclovir that the reduction in pH and the reduction in solubility was the primary mechanism. But I sort of wonder whether there were additional mechanisms that had any importance for any of these drugs. So much of this was sort of a black box approach. You're showing how well the software works, but an understanding of which mechanisms were most important for which drugs would have been helpful.
I don't know, anybody wants to comment on that?
Yeah.
Yeah, go ahead.
Yeah. Thank you, Dr. Jasko. I think you mentioned the first presentation. The project we did is for Acyclovir. Acyclovir, actually, we found that there is a delayed of Tmax compared with the situation and the fasting condition. That could be caused by the delayed gastric emptying time. That can be captured using the PBPK model we conducted. That's one of the impact of food on this drug. So what we want to say is that we can think about the drug product. What's the reason causing the food effect? What is the mechanism of drug release? And what is the property of the drug? That's something we need to consider before we build this PBPK model, and what the critical parameter we need to pay attention. Thank you.
Yeah, I think Dr. Jasko touches on a very important point here, and I think it's also aligned to what Arian was showing us with the IQ study, namely that the level of confidence seems to be related to the mechanisms of the food effect, and where those mechanisms are linked to factors which we have good in vitro tools, such as biorelevant solubility, then the confidence is higher. But there are other mechanisms where we don't yet have good in vitro approaches. Did you want to add anything, Arian, to that?
No, you explained that really well. But yeah, it's important that one of the key conclusions from our work was that the mechanism of food effect is actually very important, and we have a figure in our umbrella manuscript that highlights the different mechanisms that were involved in the molecules that we looked at, and where we had the higher confidence versus moderate versus low confidence. Like Neil mentioned, there are cases where we see very clear correlation with high confidence, where we have in vitro methodology that are well developed and well understood, and where we have that in vitro to in vivo correlation established. So that is a very good point to bring up.
I think Lucy wants to comment on this. You have to come off mute.
Yeah, thank you. Yeah, can you hear me okay?
Yes, we can. Thank you.
Okay. I want to ask a follow-up question to Dr. Riedmaier. So I'm really impressed by the IQ study. So you actually did your analysis on 30 compounds. So I'm wondering, like, based on your analysis, did you, I'm sure you emphasize the mechanism, which is very important. And also, did you incorporate the other factors such as formulation, such as the other factors which are important? So if you have all those factors, put them together, this will actually be really helpful for us to develop certain type of so-called the decision tree that can help people understand, okay, in this situation, this may be high risk in terms of when you use this model to predict, but in other situations, we actually have greater confidence. So I'm wondering whether that's something you guys are planning to develop as the next step.
Yeah, thank you for that question. We have not included formulation as a consistent parameter that we tracked in the work that we did initially. However, like I said, that is a follow-up work that we are doing, looking at formulation changes and predicting food effect with formulation change. In the initial work that we've done, we have tracked a lot of other factors in addition to the mechanism of food effect. Most of them are related to the physical chemical properties of the compound, and also the BCS classification, which is kind of a collection of that information. So we have looked at some of those phys-chem properties, and like we mentioned at the beginning, we found that the mechanism of food effect was something that really brought all of that together in terms of where we have confidence. When we combined the mechanism and the BCS, it kind of gave a bigger picture of where we have high confidence versus moderate versus low. So that is something that we've summarized in the umbrella manuscript as guidance around where we expect to have higher confidence when we use PBPK modeling for predicting food effect versus cases where we need to apply some caution. So we have looked at that, and the next step will be to also analyze formulation changes. We have not looked at food type or doses because that's something that wasn't consistently available to us. So that's still something for a future direction of this work.
Okay, can I ask another follow-up question? I know,
sure.
When you do your analysis, you present maybe nicely high-level results today. Just wondering, like, when you comparing the prediction versus the observed, did you only compare, like, say, Cmax and AUC? Did you also look at the time course of the PK profile, such as the delay of the absorption or the other parameters, such as the lag time in terms of absorption? Did you look at those parameter comparison? Thank you.
Yes, yeah, that was a very important point for us while we were doing this work. We not only wanted to look at the PK parameters, but we also wanted to make sure that we're capturing the shape of the profile correctly. So we did two separate types of analysis. We did a PK parameter analysis where we looked at the specific fold difference in terms of the AUC and Cmax, so observed to predicted AUC and Cmax ratios. And then we did a secondary analysis in terms of the shape and the fit of the curve of the profiles. So we were very strict in terms of how well the overlay looked, and you know, if there was a lag, that we captured that, and in terms of the Tmax, that we captured that well as well. And that was another criteria how we defined the confidence in terms of our prediction. So unless we had both the overlay as well as that ratio that was correctly captured, we did not assign that as a high confidence case.
Okay, Neil, you want to ask the next question?
I have a question ready, which is actually one that came in from the audience, and it's a complete different type of question than the ones we've addressed so far. It's about the software used. Arian, you mentioned that the evaluation used both SimSip and GastroPlus. And I think it was mentioned in the other presentations, the software that was used. Do you have anything to say about the different softwares, and you know, what features are different, or saying something about, you know, that aspect of the predictions?
Yeah, when we did this work, we wanted to make sure that we cover both software. So we did the exercise in parallel in both software with, I think, one or two exceptions where we couldn't get the same data set that we could fit into both of those software. So we just ended up using one for the food effect predictions. But out of the 30, the majority of them were done in parallel with both software. Our aim wasn't to do a software comparison, so I can't really comment on, you know, where one software, specifics of where one software did better or worse. And we didn't really see a very specific pattern in terms of that. So there's nothing that that was very obvious to us in terms of one performing better than the other. We saw consistent outcomes with both software. But like I said, we didn't do a systematic analysis of the software performances. We do have some details in one of the follow-up manuscripts that I cited on my slides that go into some of the details. And I did mention some points where we have proposed improvements to software capabilities in the versions that we assessed. But again, that was really consistent across both software. So nothing specific that I can comment on.
So one quick question, Arian, kind of following up on that. I mean, is there an attempt to kind of harmonize all of the various model input parameters used in different softwares, you know, and will this have any impact on the outcome of that?
Yeah, I mean, that's a good point, and I don't really, I don't know if I can comment on that right now, but like I said, as far as we saw in our analysis, there wasn't anything that was really obviously different between the two software, where one software couldn't do something that the other one could. So that was a good sign to us. But beyond that, you know, in terms of the smaller details, I think it would be good to take a look at where we saw lower confidence with our predictions and see if we can improve that in both software.
Maybe just to add, I think one aspect of the IQ study, which was, you know, special, is the use of the same standard assays to generate the input data for the 30 compounds. So I think that that's an important point in answer to your question, Partha, that, you know, if we want to build up the confidence going forward, we need to establish the best practices. And one of those would be, you know, the best assays to use for generating these input data. I think we touched on it with the dissolution already.
Right.
Yeah, and we provide
yeah, go ahead.
No, I was just going to say we do provide the decision tree in our manuscript with respect to the model inputs and how those were used in the model. So that could be used as a basis for establishing some baseline model inputs and modeling approaches going forward.
Yeah, I wanted to kind of go to Yuching. I think she's dealing with a lot of the new drug applications. So I don't know, is there any comment or position from your side as you have seen this? I think you are in mute, maybe.
I think I will answer this question. There are many different angles to answer this question. So we have several items embedded in this question. First is the generalized, harmonized, the input parameter. And the second is the harmonized in terms of the software. I actually think they are slightly different. So when we're talking about the input data set and the input parameter, they are two separate issues. What that mean? That means for some data set which can be directly used without much modification as a model input parameter, I think those are in a way is standardized the in vitro assay or data set format. But in terms of the model parameter, that means even though you come from you're using the same reference and the same data set, because of your model structure, because of what kind of assumption you made within a model, the parameter itself will be different. So in this case, I think in terms of the harmonization, from the experience we review in the new drug, we are more emphasized in terms of provide a very clear reference in terms of how you build this model, which means in terms of model structure and the model development, what kind of data set to establish those connections. Then we also focus on the output part, which is on the validation part, which will not only validate in terms of the so-called performance, but the most importantly is through those validation, can you verify the key mechanism which is relevant to your purpose, and in this case will be related to your oral absorption and the food effect. I think my experience in terms of harmonization, we need to, I think we should standardize the in vitro assay and how those in vitro assays related to the use of the model. But in terms of the harmonized model structure and the equation, right now during our experience, I think more importantly is to provide a detailed information on how you build and validate your model. Yeah. Thank you.
Thank you. Neil, over to you. I think, Neil, you are on, or if you're speaking, you are on mute.
I'm sorry, I slipped into mute there. Another question about food effect relates to the type of food, right? The type of meal. And we all know about the standard FDA breakfast, but I wonder if any of the presenters would have comments about, predicting different meal types in this type of approach. Maybe, Arian, do you want to start with that?
Yeah, sure. I think for us, it was very important to look at the different meal types. That's an important question, just to see how well we predict low fat versus high fat meals. We found that we weren't really able to do that right now with the software capabilities that are available to us. We didn't, again, do a systematic analysis on this, but it was just, we didn't think that there were enough parameters to populate the different models, the low fat versus the high fat models. So right now we're really just capturing something similar to a high fat meal. We're not able to distinguish between the different meal types. So it's definitely something that's of interest, and it's something that we'd love to follow up on. But I think it's something, it's an area where the software needs to develop still. And once that happens, it would be good to do a more systematic analysis of how these models perform in the different with the different meal types.
Fang, I think you maybe would like to say something on this.
Yeah, so for the food type, actually we know different food type will have different impact on the GI physiology. Like the high-fat meal, they may increase gastric pH more than the light-fat meal. So yeah, it's depending on the purpose of your study. For example, if you are evaluating the food effect, maybe you want to use the high-fat meal. And if you intended to just mimic the just the FDA standard breakfast, some with the light fat meal, and then or standardized meal, then you need to consider that. That's really depending on the study that you want to mimic. Yeah, and then use that to into your PBPK modeling. Thank you.
I mean, so maybe just for my understanding, I mean, having the high fat, high calorie meal, which is sort of the, worst case scenario situation. I mean, I don't know whether, right now the models are predicting that scenario and whether that's then applicable to, and whether that has sort of impacted by different drugs and the formulations are impacting that. I mean,
yeah.
So.
Yeah, for example, for if we want to see the pH DDI potential impacted by the PPI, so what gastric pH we want to use. For the high fat meal, we may use like around the pH 4 to 5, but for light meal, and we may use like 2 to 3. That may predict differently on the PK if the drug is like weak-based drugs, and or if your formulation include different acidic excipients in the formulation, that different gastric pH may have different impact on the PK of this type of drug. That's what I mean. Thank you.
I think we're moving towards the end, Partha. I don't know if you wanted to round up with your.
Okay.
Last...
Yeah. No, thank you. Again, this is a very informative, panel discussion. One of the things, as regulators, I think, I have a question, so for example, I mean, if you get to the scenario of starting to waive Fed BE studies, based on overall PBPK modeling and virtual bioequivalence, then, we probably will take a staged approach. And given the state of current knowledge, what kind of drug products and API or product attributes you would envision where risk-based waiver of Fed BE studies can be potentially implemented? I know it's a very sort of a global question, but wanted to kind of hear from the industry a framework where we can start, addressing that, head on. Go ahead, Rebeka.
Maybe I can start. So I think that Anita in her presentation gave us a good overview where she thinks how this approach would be feasible. So drug products where we have IV data available, drug products where we know the mechanism of food effect, I also would add that it is crucial that we have enough clinical data to develop and properly validate the model, and that we understand what is happening during the fasted and fed state, what is affecting the differences we see, and also that we correctly incorporate the differences between the test and reference formulations into the model. I think that would be a few points. I wouldn't say I would focus on a BCS or a specific drug, but the overall view and the collective data we have for the model to develop and validate. Maybe just another point to consider is this in vivo variability, if our model is able to capture it based on the data we have.
Anybody else? I mean, I guess so. My understanding is that you're probably more taking a case-by-case approach, depending upon what you have, what information you have, which is again, it's much harder to implement from a regulatory perspective. I don't know whether Lucy, you want to make any comments?
So, Partha, I think this is a really hard question, right? So this all depends on how well you understand your drug product and the how much you understand the mechanism that the food is having how the food is having an impact. Also after you understanding all this, you have good in vitro, in vivo data, then you have to build a really the PBPK model with high confidence. So only after all those steps, you will say, okay, now I'm confident enough to use my model to predict the fat PK or the fat BE. So, back to your question, should we do it case by case, or should we have certain, that's the question I was trying to ask Dr. Riedmaier. It's like, is there a certain decision tree that we can follow? Okay, in this situation, it's a rather higher risk for us to use it right now, or in this certain situation. For example, take it simply that if it's an IR formulation with PCS class 1, so probably there's a relatively simpler formulation, easier compound, probably we have greater confidence. But on the other situation, if you have more complex formulation, then you may not well understand the mechanism behind. So in that situation, maybe we have to be a little bit more conservative. So to me, this is really how much you understand your product and the mechanism behind.
Yeah, I would also kind of add, I think it's also dependent upon a lot of the patient factors, that needs to be considered, and so the context of use becomes very important in this regard also. But I think it will be nice to kind of, as I said, take a staged approach and trying to address at least the low-hanging fruit where you have the most confidence, and move forward. And as you move forward, then you will collect more information that will make you more comfortable. Because as regulators, oftentimes, we don't follow the best case scenario. We always, worry about the worst case scenario, and which might be a minority of the cases, but still we have to worry about that part. So it's a very fine balance, between, what we know, what can be done, but at the same time, can we implement an approach that works for most of the cases. I think any last comment from anybody?
Thanks, Partha. Thanks, Neil. Thanks to the whole panel. It is this discussion of food effect leads us to the so-called lunch break, at least here on the East Coast. And we will reconvene with session 3, challenges and successful cases for oral PBPK, in 45 minutes. That is at 1:15 p.m. Eastern time. Have a good lunch break.
Thank you. Thank you, everybody.
Thank you very much.
Bye.
Thank you. Bye.